All right, so welcome to the Austin Python Meetup. I think it's our 8th meetup of 2021. Thank you everyone for coming from Austin and from around the world. Today we have two great talks. We have Maciek Swat, who is going to talk about using Python to democratize the virtual tissues modeling. That should be a very, very exciting talk. And our second talk is going to be given by Charles Lindsay, who's going to talk about Bayesian statistics with Python. No sampling necessary. So so Maciek and Charles, feel free to introduce yourselves. I'm not going to do the introduction. You feel free to introduce yourselves the best way or the way you like to. And then Maciek, the floor is yours.
All right, thank you very much.
Thank you.
Let me share my screen. And there we go. So, right. So my name is Maciek Swat. I work at Inscripta. So this is the company that does devices I was just showing before for genome editing. So we are building essentially the type of device. Think of it as 3D printer for living organisms, where you put, for example, bacteria or yeast with one set of genome, and then you make precise genome modification, and you get those modified organisms on the other end. So that's what I'm working on right now. But previously, I have spent 15 years working at Indiana University, where I was doing computational biology. And as Jacob mentioned, we've met at this MSM multi-scale modeling consortium organized by NIH in Bethesda a while ago. So I'm going to talk about virtual tissues. And the first question that comes up naturally is, what are virtual tissues? So virtual tissues are essentially, I would say, computer infrastructure. And when I say infrastructure, I mean models, software, hardware sometimes, that allows you to, let me actually make it smaller, that allows you to simulate tissue development, biological tissue development, homeostasis or disease, and interpret and compare results of the simulation with experimental results. So I'm going to show you one such example. So this is the work that we published 10 or so years ago, and it's a tumor model. A tumor is a particular type of tissue, right? Where, so here you have a vascular structure with blood flow, and here you have tiny bit of tumor, of cancer cells. So what happens is when we run this model is the tumor initially starts growing, right? It gets nutrients from the surrounding blood vessels. But as the tumor gets bigger, the inside of the tumor doesn't get nutrients because there are no blood vessels inside the tumor. So what happens is that those cells which are inside, they are almost dying. But in order to prevent their death, they are sending signal, and the signal is called vascular endothelial growth factor, which essentially is a signal to stimulate blood vessels growth, so that blood vessels can sprout from existing vasculature, invade tumor, and supply nutrients. And this is what we are, and let's see how the simulation progresses. So those purple segments here are the new blood vessels that are being formed in response to those cells that are inside the tumor, and they are deprived of nutrients. And as the blood vessels, new blood vessels grow, they surround tumor, they supply more nutrients, and tumor grows. And this is typically what happens with cancer, right? This is what gives rise to this uncontrollable tumor growth. So the natural question then is, what would happen if we could suppress somehow formation of the new blood vessels? So then we can build a simulation. And notice that, when I say we can build a simulation, those simulations are much faster. Building them and running them is much faster than doing actual experiments. So let's see what happens. So this simulation is exactly like this one, except right now we simulate the action of the drug that prevents new blood vessels from being formed. So notice what happens. The tumor was in the middle. There are no new blood vessels, but the tumor grows. But notice how tumor migrates from the middle of the lattice to the side of the lattice, and it aligns itself along the blood vessels. Why along blood vessels? Because this is where the nutrients are. So by doing this simple, well, I don't want to say simple, it was actually, it's one of the most frequently published cited papers by group that I was working with. By doing this simulation, we essentially simulated the scenario where when giving drug that suppresses blood vessel formation, we actually are making tumor more migratory, or in other words, the tumor has more metastatic potential. So this is sort of like interesting motivation why you want to build virtual tissues, because you can set up those virtual experiments and you can study those what-if scenarios without doing costly experiments. So that's what it is. So where did the term virtual tissue came from? Well, it was coined, and I think I will credit Dr. Thomas Knudsen, whom I know, and he's a toxicologist who works at the EPA. And I think EPA essentially coined this term. And the reason why they did this was that at some point when European Union formed, one of the mandates behind European Union was that European Union should issue toxicity recommendation behind every single chemical product that's on the market. And it was a big, and it is, big technical challenge, right, to test and screen every chemical compound against toxicity, against, how it affects human tissue function. So one of the ways in which you could, short circuit this, or solve this problem would be to look into computer simulations, right? So instead of doing real experiments, you could design, you can build a set of computer simulations where you can test those chemicals. Now, I just want to be explicit here. This is extremely challenging task, and there's no way, we are not there yet. We can simulate part of it. We can simulate action of the certain chemicals, but I don't think that we are right now in the position to screen thousands of chemicals just using computer simulation. This is way too challenging, and computer simulation is not there yet. So, but this is a little bit of history. And one of the projects that EPA is essentially pursuing is virtual embryo, where, they build embryos, so they start, essentially from single cell, and they simulate, they have models of embryo development, and then they simulate how different chemicals perturb normal development, and they see what type of defects those such embryos develop. Okay, so let me go to the next slide. Okay, so besides virtual embryo, there's also virtual thyroid, and then there are virtual other organs that you can simulate. And let's look at this picture here. So how would we actually start modeling virtual tissues? And I showed you a simulation of cancer, but how, if I were to ask you to build a virtual tissue simulator, what would you do? Well, the way I like explaining this is this. If we look at Yanis trying to shoot, free throw, what we can do, and most of us can do this, or everybody here, we can build a model, mathematical model of basketball trajectory, right? Just given the initial velocity and the height at which the basketball is, we can simulate what's going to happen with the basketball, and more precisely whether the free throw will be successful or not. And the reason I'm showing this is simple, because simulating virtual tissues is very much like simulating the basketball trajectory, except right now, instead of computing basketball trajectory, we will be computing trajectory of every single cell in our simulation, right? So the task of building virtual tissue simulation is essentially a task of tracking position or trajectory of every single cell. And why do we want to do this? That's another question. And the reason why we want to do this is because many tissue defects or diseases arise because cells end up in the wrong place at the wrong time, or they are doing wrong things. Example, well, I showed you cancer simulation. You have cells that have abnormal behavior and they're in the wrong place. meaning, they occupy space of your body that they shouldn't be occupying. So that's the reason why we are building those models. And how do we approach this? So essentially the goal of virtual tissues is slightly different than the role of genetics. In genetics, if you look at human genome, you can say that humans have, let's say, 20 to 30,000 genes. That's a large number. And each cell has those genes, and those genes can be turned on or off and so on. But if we look at a single cell and think about cellular behaviors, so if you ask ourselves, what do cells do? They typically do very few things. So what cells can do? Cells can grow, cells can divide, cells can die. If certain cells don't die, well, that's essentially cancer. Cells can stick to each other, which is very important interaction between cells. They absorb and secrete chemicals. They can differentiate, they can change types. For example, they can change from stem cells into differentiated cells. And there are a few other behaviors, but this list is really short. There's like, 10, maybe 15 behaviors. Compare this to thousands of genes. So naturally, yes.
I got a question. So what are like the senescent cells? I heard about like there's cells that they're like kind of like, yeah, not quite alive, but not dead either. Like would be, like where would you categorize those senescent cells as?
Yeah, so I don't think that we are, that's a good question. So instead of actually rather ask, so let me rephrase this question. What I would do about senescent cells, I would essentially introduce type senescent cells, and I would describe what type of properties they have. For example, are they growing, are they dividing, or are they dying or not, right? Or how strong they adhere. So I will try to, for every single cell type, whether this is, senescent cell or if we're talking about cells in your liver, in your kidney, in your retina, I would try to phrase the behavior, the set of behaviors that describe the cell in terms of how well or how fast do they grow, how fast do they divide, and so on. That's what I would do. Does it make sense?
Yep.
All right.
Thank you.
So that's where we are. So now, I showed you just one particular simulation that we have built of this tumor, and I will show you more, of course. But there are many mathematical methods in which we can represent the tissue motion within the tissue. And there are, of course, trade-offs between computational efficiency and accuracy. And I just want to say that there is no one-size-fits-all solution. So very often when you have a problem, when you have a particular problem, you're working with biologists or a doctor, you want to choose the right method. And let me actually give you an overview. And I might actually skip some of the methods that are out there, but this should give you a good idea. I don't know how to close this guy, but anyway.
What metrics do you use to decide, accuracy, of your representation?
Oh, you mean, that's a good question. So you mean, so essentially, is the question how, after we run simulation, what do we do? How do we compare it to?
Yeah. How do you see how good your simulation actually is? I mean, computational efficiency, like how fast is it? Those are pretty simple, but
yeah,
how do you measure the accuracy from one to the next?
This is very good question, and this is excellent question. This is one of the problems where there is no one good solution. And I will say even more. Sometimes, like if you look at those models, sometimes you might have to simplify. It's not like there is one-to-one correspondence between cells that you represent in your model to cells in the real picture. So what type of metrics we can use? Well, we can use metrics that describe vascular structure. We can use metrics that describe average cell motility. So in other words, we can, a microscopic image, we can track what is the typical displacement of a single cell as a function of time, how far it travels. And of course, and then we can compare it with the simulation. This is how you actually get your time constants. But
yeah,
in general, I would say this is open topic, and if you have some ideas that could be used, I mean, I'll be super happy to talk to you offline.
Yeah, well, I don't have like that much spatial experience, so that was why I was kind of interested. Because it is a great
question. It's a great
problem.
question. Yeah. Yes. But anyway, so
it's actually it may be a good time. It's like if Maciek told this before, but those models are not at the level of maturity you think about. This is still what he's talking about is something that's still developing and will develop for a long time before you can actually see it. Because those things are, as far as I see, and I've been doing this for a long time, and Maciek even longer than me, then those things are not 100%. They are far from 100%. And what you're talking about is validation.
Yeah.
Like Maciek said, it's not only an open problem. There's like many paths, and it's not very good right now. This is the real truth.
I think that's fair. Because if there was a good
there are like so many different techniques, you haven't like established like a gold standard or anything to compare to.
We are far from it.
Unfortunately, yeah. I mean, in some cases we could do some we are doing validation, because otherwise it wouldn't make much sense. And I will actually talk about it. But it's challenging problem in general. So going back to the presentation. So there are many methods, and the one division that I like how I separate those methods is methods that treat single cell as a point in space. And there are a bunch of methods starting from cellular automaton to center model. So this is more like game theory based. Center model is a physics based. So we have spring-like interactions between cells, and they essentially drive pattern formation.
I think you're mute, Maciek. Just turn your mic to mute.
Sorry. And there are other methods where you explicitly represent cell shape. I like those methods. And why do I like those methods? Because A, they look closer to how real biological cells look like. B, there are many biological phenomena, which I'm not going to talk about, that actually arise naturally in those models, versus if your model treats cell as a point in space, you have to do a little bit of faking. So those methods that represent cell shape spatially, explicitly, are my favorite, but of course you pay the price in terms of, efficiency. And there are different approaches. I'm not going to go through all of that, but just to let you know that there are many people working on those problems. I have to do this. Essentially, I want to show you virtual tissues in action. So I'm going to show you how we approach the problem of modeling polycystic kidney disease. And polycystic kidney disease is a disease of kidneys. So this is your normal kidney. And if you have polycystic kidney disease, your normal kidney, which is 10, 12 centimeters, grows to the size of football and is embedded in your body. So you can imagine that this is problematic, right? And it's lethal in the end. So how did we approach this problem from virtual tissue perspective? Well, we know already that all cases of polycystic kidney disease are due to genetic mutations. We know, and we know exactly even which genes are mutated. But what we don't know is, okay, you have mutation in genes, but how do those cysts form? Like, what is the actual mechanism? So we had some ideas that cell adhesion might play the role. So we asked the question, let's build a model and let's see if deficient cell adhesion is sufficient to initiate cyst formation. The next question we can ask: how big perturbation, how big deficiency in cell adhesion you need to start cyst formation? The next question is, can you control this loss of adhesion using drugs? If so, at which dose? And then another question was, how about cell proliferation? If you have too much, cell proliferation, does it also contribute to a cyst formation? So those are the good questions for our virtual tissue model. And this is exactly what we did. There are exactly a few questions that our virtual tissues cannot answer, that we cannot answer exactly how at the molecular level things happen, but we can look at the cellular behaviors and see if they really affect formation of cysts. So we have built this model, and the work was done by Giulio Belmonte. He did fantastic work. So this is a piece of renal tubule. So this is a tubule that's in your kidney. And what we are doing, we are knocking out adhesion molecules in one of the cells. We're just like lowering the adhesion. And those cells they're forming epithelial sheet, and they're under tension. So if we make one link weaker, this is what happens. It's enough to initiate cyst, right? Just knocking this out. So that immediately suggests that, really cell adhesion is important in this disease. So the next question is, can we actually come up with the drug, or can we somehow control cell adhesion and make sure that we can control this disease? So that's indeed what the team did. So the team was composed of, nephrologist, Bob Bacallao, James Blejer, who was professor, Sherry Clendenon, and Giulio Belmonte, who did all those fantastic simulations. And long story short, I can tell you that those simulations, those virtual tissue simulations that were kind of, the nephrologist who worked with us was very skeptical. Later on, he was a total convert. They led him and Sherry Clendenon to discover potential mechanisms in which they can control cell adhesion. They found existing drugs, and they started, trials. So that was actually, I think, one of the big successes in our lab. And I'm sure that there are many other labs, which, I showed you a few slides before, that do also fantastic work, for example, in sickle cell anemia, that have similar success stories. So this is what virtual tissues do. Now, I just want to spend a few minutes talking about platform that we use. So the platform that I use, the platform that I was working on, and I helped develop, I developed most of it actually, is a platform called CompuCell. And CompuCell, for me personally, I was always jealous of MATLAB, because I think that this paradigm where you have this one environment that solves many problems is good. I'm not a big fan of MATLAB. I like the idea. I coming from Python world and then jumping to MATLAB was painful, and I actually stopped using MATLAB because I think Python is much better language overall. But the idea behind MATLAB as a problem-solving environment is alluring. Because if you give users, and I will show you, demonstrate quickly, if you give users user interfaces, if you give them fast computational engine written in C++, if you give them scripting language, which, in this case is Python, if you give them model editors when they can quickly configure simulation, you have recipe for success. And I don't want to boast too much or, say that we achieved the success, but what happens was that, when we taught CompuCell workshops originally, it took us full day to teach people how to do basic simulation. Right now, basic simulation in CompuCell is being done within 60 seconds. And essentially all you have to do, there are a bunch of screenshots, and the styling of those dialogues is kind of weird. It comes from old Windows, but don't pay attention to how it looks. I would pay attention to what it does. Essentially, those 3 screens allow you to build skeleton of fully functional simulation within literally a few clicks. And why is it important? Because in typical scenario, when you build virtual tissue simulations, 80 to 90% of code can be auto-generated. It's all boilerplate, right? So what's not boilerplate? Well, and so this is actually, after you build the simulation, you get, simulation code. So part of the simulation is described using XML, which I don't like, but it comes from the fact that, 20 years ago when you were writing NIH grants, if you had XML word in your grant, you were getting brownie points. So I think this is the origin of why we are using still XML. But most importantly, the essence of the CompuCell simulation is Python scripts. Are Python scripts where users write code that any type of code that can interact with simulations objects and do whatever they want at any time. So this is essentially the architecture. Again, high performance C++ core, Python scripts, and user interfaces. And typically what happens within the simulation is that, the simulation is divided into steps. So you run your C++ engine for a number of micro steps, and then you jump to Python. You adjust properties of all the simulation, of all the objects. And by the way, we represent our cells as a collection of voxels on a 2D or 3D lattice. So those are 4 different cells. And then we use energy formalism to drive cell dynamics, essentially to perturb boundaries of the cells and drive system to lower energy. So this is energy-based formalism, which can be translated into force-based formalism, but I'm not going to go into details. The most important thing here is to realize that you're jumping between C++ and Python code. And that's what makes a huge difference for you as a user. So typically, so this is the script. Typically what we do, the canonical pattern that we always encounter is this one. You iterate over all cells in your simulation and you check, if cell type, it's essentially, you can read this. If certain type, or if cell type is condensing and time or Monte Carlo step is less than 1500, you're reading concentration of the chemical field at the center of mass of the cell, and then you are increasing its target volume by the fraction of the concentration. So what happens is that as a result of this simulation, cells here, so this cell secretes chemical, and those cells grow in response to the chemical. So if the concentration of chemical inside here is high, the cell grows faster. So this is how you would normally start building your tumor model.
Would that loop, can it be parallelizable? I know you mentioned using GPUs and stuff like that.
Yes. So we have parallelization, and there are multiple levels. So I think you get the biggest bang from parallelizing by using parallel PDE solvers, partial differential equation solvers. And we have diffusion solvers which runs on multi-core, on CPUs, or on GPUs. We would like to have more of that. I mean, we're just starting a little bit out of steam in terms of development and maintaining all those GUIs. But yes, and then the core algorithm for simulating cell dynamics is also parallelized, but it's parallelized only using OpenMP. Would love to have it on multiple nodes as well.
Gotcha.
And going back, it doesn't really make that much sense to actually port this algorithm to GPUs. It will run faster, but then certain classes of the simulation, certain important class of simulations will not be able to run. And I'm not going to go into details, but it is possible, but there will be big trade-offs.
Yeah, gotcha.
So
not everything is.
So this is essentially the end of my talk. I mean, one if I can ask you a question, you guys, some of you might be actually experts in high performance computing. If I could ask you a question, if there's any way to speed up this pattern where you iterate, so this cell list is a C++ container of objects, of cell objects. And we are iterating very frequently throughout this list. And this is the source of one of the biggest slowdowns. If we can somehow speed up this part, we could have much better performance. So, this is a question to you. You don't have to answer it right now, but, if you happen to know solution or can point us in the right direction, we will be very grateful. And let me actually briefly show acknowledgements. And I want to just take 2 minutes and show you the 60 seconds and show you that I can build the simulation within 60 seconds. So let me thank to James Glazier, James Luca, TJ Sigo, Abbas, Julio, Sherry, and Bob for all this work. And of course, I want to thank you for inviting me. It was fun giving this talk. And let me now briefly show you how you build the simulation within 60 seconds. So you open this Twedit editor. It's all Python, by the way. The editor uses QScintilla engine, which unfortunately is GPU. You open new project. You call your simulation. I will call it Austin demo. All right. The simulation will be composed of Python and XML. You click next. Your lattice dimensions are predefined. Number of steps is predefined. And initial cell layout is predefined. I'm going to jump next. I'm going to add 2 cell types, A, B. Those are my 2 cell types. I can add C if you want. Not a big deal. Continue. Let's add diffusion solver. Let's introduce new field. I'll call it FGF. Oh, GFG. Okay. We'll do. And then we're going to select cell interaction or cell behaviors. I want contact, and I want this guy, volume. Essentially, I want my cells to have certain volume constraint. I want to hit this. My simulation is done. If I double click on my simulation code opens, if I want to run it, I should be able to run it. I hope it runs. I didn't rehearse this. So this is your skeleton of running simulation, right? It didn't really take us more than 60 seconds. Maybe it did, because I have to talk you over. But now, why Python is important? Well, Python is important because, for example, if I want to implement cell growth, I can do it really quickly. Sorry, let me actually pause this. I'll show you just really quickly, and I'll stop soon. Cell dot. Let's actually make it faster. Sorry, 02, and let's do this. If cell dot type equals self A. Okay, let's do this. Save, and let's rerun it. So what should happen now is that if cell type is 2, one of those cells should start growing. I'm not sure if, let's switch back to they're not growing. Can you imagine that? That's a little bit annoying. Let me actually, oh, I know why. I'm sorry. They are not supposed to be growing, but I will make them grow. So essentially, I have hard-coded target volume to be fixed throughout the simulation. Now, I'm going to make this, or let me actually, oh, by the way, when we are writing this code, when we are writing any type of iteration, we have those code assistants where we can essentially plug the code that iterates over all cells. And this significantly speeds up the development of five, and then I'll do this and lambda volume. So this is equals, sorry, lambda volume equals 2. So So lambda volume is essentially the strength of your, how strongly cells should right? So now you can see that those blue cells are growing indeed, right?
Yeah, they're overtaking.
Yeah.
They overtaken. Now, if you want those cells to divide, I'm just gonna, you know, this model is sort of like silly, but you can easily add new module called mitosis, and you can essentially have cells getting divided. And the only reason I'm showing this right now is just to demonstrate that indeed, as I mentioned, Python, by using Python in the right way, you can democratize access to virtual tissues in a way that people don't have to know. They don't have to know. It's the, for example, C++ or C or some compiled languages in order to build sophisticated simulations.
Mm-hmm.
This simulation works exactly as expected, and the reason why it works, I'm not going to actually go into details right now because we have another speaker and I don't want to steal his time, was that we have plenty of blue cells, but when the blue cells divide, they turn into green, and greens don't grow.
Mm-hmm.
So I'm going to stop right here. I think that's enough. I hope that it was useful for you. And yeah, thank you very much.
Yeah, this is fascinating. Cool. So is it like open source, or is it something that...
It is.
Yeah.
It is open source. And if you go to computer3d.org, we just finished the workshop. I wouldn't open the, well, I mean, the quality of source code improved, but, keep in mind that it's an academic project that we started a long time ago, and, we didn't have as much time to clean up the code as we want to, but it is open source. And if you or anybody has some ideas for even tiny improvements to, either performance or how you validate against experiments, please contact us. We'll be super grateful and would love to work with you.
Yeah, definitely. This is very important for probably for the whole world, right? Like for everybody. So I think this is fascinating and also very important work that you're doing here. So this is amazing.
Yep.
And you're continuing to actively develop it too?
Yes, I do. And actually, there is a new lead developer who is very, very good, TJ Sigo. So the team is active. Yeah, I don't spend as much time as I used to, but this is a very much active project.
Yeah, cool.
So, okay, I'll stop sharing, and yeah, let's jump to the next speaker.
We have questions. Don't go away yet.
Okay, no, I will stay.
There you go. It's like I didn't jump in because this is fascinating, and people already figured out that this is important. But even on this call, I know people who were like, I looked at the names. I know people who were interested in biology for a while and came from biology and started doing some other things like machine learning.
Yeah.
And you were mentioning something about the ability to actually parallelize some of your work. In the Python meetup here this year, we had a presentation from a company called Coiled. They now give free computing time up to 100, I believe, something. I asked them, it might have changed, but you can actually go and run your simulations on their parallelization thing. They are based on something called Dusk. Very easy.
Dusk.
Yeah, so if you can plug in the problem you said about the high performance computing, check out the Dusk. If you can run on Python 3 in the newer stacks, then you should be able to actually parallelize whatever you're doing pretty easy. And the company that does this is Coiled. They actually have presence in Austin. The CEO is in Austin.
Yeah.
So
I know Dusk. I know Dusk. It's one of the things that I was actually thinking, but again, about parallelizing at least PDE solvers. But obviously, there's only so much you can do when you have other job.
I know, but Dusk is your solution, and now Coiled gives you, while they're in beta, or I don't know exactly what the deal is, check out the website, go and check, they might give you even free computing time to actually do the parallelization. So if it might be worthwhile talking to them, and you can, if you look at Florian later will show the presentations that we had this year. Florian had has a link. He will show you what they showed us. I think it was April or March this year.
Yeah.
They showed.
Yeah, I'll send you the link to the YouTube channel. I think if you remember Cliff Kerr, I think he used to work for, or he's working for Bill and Melinda Gates Foundation or some in Washington.
Cliff Kerr is a different type of a disease model.
Yeah,
like
but
he was talking about disease models.
Yeah, he did a lot of optimization. You remember he talked about optimizing their code as like they optimize it like it's like some enormous optimization that they've done. And I forget exactly what they use. I think they put everything in columns. I don't know if it's the same problem, but I heard him talking about optimization. I'll also send you that link to his talk, because he talks, again, I don't know if the optimization you're looking for that you just mentioned is similar to his, but it might be interesting, maybe get some ideas.
That would be great. Yeah, any leads, who knows?
Yeah.
But that's always helpful.
Definitely.
So this is one thing, and this is one thing that I noticed in your presentation. If you're looking for high performance computing, there are ways, and Python is definitely the language. Another thing is that there's something that's like you were saying that people still don't get it. You show them the stuff and they're fascinated, but it's important to know this is not flight yet. This is pre-flight. What they're doing is like what Maciek is doing is like the Wright brothers trying to make their first plane. This is the analogy they can find. Yes, there are things that are getting off the ground, but it's not real flight yet. And Maciek is kind of like very, very conservative in saying this, but it's important to know once those things start being really, really useful and enough time is invested in to make them better, then those things will go up. So our medicine, our biology, our well-being will be much, much better in the future. We're not there yet.
Yeah.
We have a long way to go.
I think that's fair. I mean, although, as you said, big progress has been made, but I kind of agree, and especially on the platform front, I can see that definitely way more effort is needed than what currently has been done. CompuCell is a proof of concept that yes, if you write software in a good way, if you make it user-friendly, it can work and people will use it, but we want to go further beyond that. So I completely agree. Yeah.
This is a vision that I believe there are several people on this meetup that share this. I noticed some names. If they want to, talk and join in, it's maybe a good time and talk to Maciek before, because it's like there are many, many people who believe in it, but it's still a big, big, big challenge.
Yes. Cool.
And Python is very important here, at least to my understanding.
Yeah, Python actually, I don't want to preach the choir, but this code started as C++ with XML configuration. And there are some approaches where people say, hey, you need to use GUIs only. And I kind of see place for GUIs, but GUI-based configuration of simulation is not going to fly in general. You need to have scripting. And Python was this thing that actually enabled CompuCell and allowed us to actually branch off and let people around the globe to use it. And how many people are using? I don't know, and I don't want to know, because this is how you measure success. If you get many people that you don't know who use your software, that means that it's successful, and we are working hard to get there. But it's been actually, as Jacob said, very important to choose the right tools for this project, and I think Python is such tool. And obviously now with all machine learning and everybody jumping into Python, it sounds obvious and trivial.
The GUI can kind of be a good starting point too, right? You can generate the Python from the GUI and then tweak that, right?
Exactly.
Yeah.
Totally.
Cool.
I mean, it's amazing how flexible this whole ecosystem is. It's unbelievable. And we are using Swig to wrap our code. It was pre-PyBind 11 that when we started, but and this combination of C++ and Swig and Swig and Python is so powerful that I would use it everywhere I could. I mean.
Mm-hmm.
There is someone who's asking a question in the chat. Maybe some of the people sometimes don't, but Roger Williamson is asking, are you doing statistical techniques like Bayesian inference or maybe HMMs?
So that's a good question. We, in general, we would love to actually build AI-based surrogates for some of our components. For example, maybe we can train neural network, and I know that people have done this successfully, to speed up the diffusion solvers. We don't need to get exact numbers. We just need to get ballpark within reasonable accuracy. And this could be the biggest gain.
You might want to go slower here and explain, because some people don't get your language.
Okay, that's a good question. So what are we using statistical techniques? Like, we don't currently know. And no, we are not using those. That said, we would like to start using neural networks, for example, to replace some of our PDE solvers, for example, diffusion solver, with a pre-trained model that's smart enough that given initial condition and given diffusion constants, it can give us solution to this diffusion equation at some later time. And this has been shown to give huge boosts. We are talking about factor of 1,000 speedups. And if you can get factor of 1,000 speedups, that would be good. Statistical techniques for in what sense would we use those techniques? Like, I'm just thinking
Monte Carlo simulation.
Well, those are all Monte Carlo simulations, by the way, that I was showing, at least the simulations of.
So you do have some statistical elements there. Monte Carlo.
Yes, we do. But for us, statistical modeling, we were talking about like statistical inference, right? Exactly. And this, I don't think we really have taken full advantage of the development. We're looking into incorporating this. We're looking for efficient exploration of parameter space, because our models have to have parameters and we don't know often what they are, so we have to sometimes find them. We're looking for ensemble modeling, see how different simulate populations or cohorts of patients or different tissues and see the response. So I'm sure there is a place for statistical modeling, except we have not taken full advantage of this.
I guess the question that really should come before that anyway is, do you have statistical data? Do you have distributions of the interactions of the characteristics that you're modeling? If you would need those.
That's right.
I suppose. I mean, you can always assume them. I mean, maybe this is a way to discover them by modeling, the way Bayesians do, with starting out with no known distribution and then discovering what works.
No, I think you're right, Roger. I think you're right, and exactly that's one of our thinking is essentially, in the absence of biological data of the distributions, right? We could actually come up with, with some artificial data and see if our assumption gives us anything that resembles, what experimental is fine. But for us, it's actually very hard, at least to my understanding, to find good experimental data a priori. So doing the artificial data first with proper distributions might be more well, be better way to go forward.
By the way, I think what you're doing is wonderful, and I applaud your work, and very happy to see that kind of work being done.
Yeah, it's I'm just reading. Yes, I'm just reading the comments from David here about protein. Yeah.
So Roger and Maciek says they both kind of aim it at something that's also important to understand. There isn't a lot of data to actually validate against or actually learn from. This is part of the bigger problem here.
Mm-hmm.
And interestingly enough, now the NIH is starting a big program called Bridge to AI. The idea is to generate data so in the future AI can do things with it.
Yeah.
So this is something that you should be aware of. Some of the answers that Maciek was giving was like the best that are possible today. We're talking about tomorrow, and all of those tools that we now see are basically the beginning of things. And in the future, they might change, or some of them will be adopted or changed to some other things. But it's important to know that this world exists and Python is a part of it. And of course, since we're going to AI, Python is going to be there for a long time.
So from a management standpoint, this could be just extraordinary, because management needs to know where to go look and what to ask. And one of since you have questions about the statistics for these various processes, that should guide what people do in the future to bring up or to generate that data. And so it's an iterative type process, is that you say, oh, I wish we had this kind of data, and somebody says, oh, well, I can produce that data for you, and off you go.
Well, this is what they're trying, and yes, this is a lot of it is experimentation, and Maciek actually says like we can do things like that. So
yep.
And I ask, I wonder whether statistics can be used for the direction of the tumor growth.
Yes, I'm sure there when it comes to tumor growth, there are many models. Some models actually don't even look at cells. You have those PBPK, meaning pharmacologically based, physics-based pharmacokinetics models, where they essentially look at the distribution of the growth of the tumor mass, distribution of drugs, and so on. And they don't even look at spatial things. But and they often will use statistics, just to quantify. And this is essentially at the clinical level. They could triage, different tumor growths into different categories and so on. But here we are taking different approach. We're essentially using physics, real interactions. We're telling how strongly cells are growing, how strongly they are sticking to each other, and so on. And so it's slightly different approach to tumor growth. I'm not sure if this answers your question.
Yeah, I'm thinking, but those things can be assigned statistical weights, can't they?
Yes, it could. But I'm just commenting.
It all kind of brings to mind hidden states that, as you generate the model, the clinical data is going to tell you what the total is going to look like, even though they can't see.
That's right.
The states that you're using internally, but eventually your model has to produce their data, and from that, maybe there'll be insights developed in the hidden states.
Yes, that's true.
your suggestions may be good. The thing is, there is a need for people to actually go and follow them, and people who are interested in doing this, the tools are available. Just need more and more people, and this comes to the point of how many people are going to do this, and this eventually comes to funding and some other issues. So
that's right. That's unfortunately very much how the reality looks like, and you know, maybe as Jacob said, you know, earlier, the number of people, this field in general is not as well represented as, for example, bioinformatics, and you know, it's understandable. Bioinformatics plays profound role. I'm actually doing bioinformatics right now in my job, but I think we need a little bit more awareness and effort from other labs. I think it's changing. There are more and more people actually appreciating this type of mechanistic modeling, what it can give you. So while statistics,
tell them what mechanistic model is, because some of them don't know.
A mechanistic model is essentially a model where you try to understand how certain things happen by understanding like basic mechanisms. How, you know, if you, for example, I have showed you how tumor becomes more migratory, migrates more when you turn off new blood vessel formation. When you give anti-angiogenic factor, you know, tumor tries to align itself. So this is essentially, you're trying to build a model that will, you know, explain this phenomena. I'm not saying that this happens in every tumor, but I think there is a class of tumors in which it could happen. So so that's type of mechanistic modeling, you know. Not sure if this is clear enough explanation, but it's a simple.
It's opposed to the stochastic models that were things random. Basically, people kind of divide stochastic and mechanistics. There is a bit of overlap, but basically it means that there is some mechanics behind the model that you can explain, as opposed to things that are kind of random. Is it the better explanation, Maciek, or?
Yeah, I mean, the way I look, mechanistic to me means explain the mechanism of, explain how something happens, explain how, I don't know, when gas prices go up, the inflation rises. Like, for example, that's the type of, that's what I mean by mechanistic model. It doesn't have to have necessarily.
How would that interact with talking about, like, okay, this is a parametric model, as opposed to like a not, you know, really vague, agnostic, non-parametric model? We're inferring everything.
So
is that kind of the divide you're thinking of?
No, well, I mean, I wouldn't, you know, I'm not a big expert on actually this taxonomy of models, you know, how I don't think it, you know, we can do those classifications, but I think what's important here is just to see what the model can do for you. Going back to parametric model, all the model that the simulations I've presented, they use parameters, right? There are real numbers, and those numbers ideally should be mapped and are mappable, by the way, to biological measurements. The problem is that often you don't have those biological measurements. So this is another opportunity. Given the model, given the lack of biological measurements, hey, maybe you can actually use your models to constrain at least some ranges of the parameters that your model has. So, you know, if that makes sense. All right.
All right.
So I think I'll stop sharing my screen and let's jump to the next talk, all right?
Well, thanks.
Oh, we have to thank you.
And thank you.
Oh, yeah.
Thank you so much. This was fascinating.
Yeah, that was really, really cool. So
It was a pleasure actually talking to you all.
It's fascinating. Thank you so much, Maciek.
Sure.
Appreciate it.
Yeah, thank you for sharing.
and so Florin, you're keeping track of you know, like the links he's given us and stuff like that. So
Well, it's gonna be.
like,
I'm recording it right now, so I'm gonna post
okay.
everything. Yeah, everything's gonna be on our YouTube channel. So I'll post again. Like once I post.
Okay, gotcha. And maybe like in the comments for the video, we could put the website for CompuCell 3D, stuff like that.
Yeah,
Awesome.
yeah, we can do that. All right, I guess it's your turn.
All right.